screening as well as development. The next speaker is Neta from TEVA. For me, Neta is always representing ALS, so I hope that she will bring this
now representing many rare diseases, not
yes,
just ALS.
now in TEVA.
Yeah. Okay. לא כל כך... אוקיי. סשן 2. כן. So thank you, everyone. I'm honored to be here. As Udi mentioned, I used to be in these conferences as representative of nonprofits. I'm very happy to see that there was a great representation of nonprofit, not by me. So I'm here representing TEVA, and I think, as far as I can see, we're the only industry member in this conference, so we're honored to be the member. And I think it's very important to understand the importance of engaging industry for drug development for rare diseases. So we all need to work together, academics, patients, clinicians, and industry, to get a cure for rare diseases. So why would we want to focus on rare diseases? I'm going to talk about why we want to talk about it, what are the challenges specifically for rare diseases, and how hopefully we could start to overcome them together. So why would we want to focus on rare diseases? There are two answers for that. The first one is that we have commitment to patients. As other people have said here before, rare diseases are not so rare. Each one of them is rare, some of them extremely rare, but when combined together, they're extremely common. 1 in 10 people has rare diseases, some of them of the most debilitating diseases existing right now. So we have a commitment to solve them, and I think everyone in this room right now has that commitment. And the second thing is that I think we could learn from rare diseases, and we hope to learn from rare diseases, because eventually we want to progress to solve as many diseases as possible. So a little bit about commitment. So TEVA has a commitment for rare diseases. If you look at our pipeline, you can see that rare diseases are well represented across it. So you could see we have in phase 1, we have treatment for Tourette. In phase 2, we have multiple treatments for Huntington disease. And we already have in registration a treatment for Huntington disease. We also have a lot of treatments for rare diseases that are now in preclinical stages that I didn't want to list all of them because there are several of them. So just a little bit about the commitment. Eventually, all of the work that is being done in this room would ideally reach this stage, this registration stage. Because at the end of the day, we don't only want to identify the reasons for rare diseases, we want to develop them, and eventually we also want to reach the patient and give cures for the patients. So another reasoning, so just to go over the reasoning to focus on rare diseases. So rare diseases are also fundamental to understanding common diseases. So we don't only want to focus on them for the benefits of the patients with rare diseases, we also want to focus on them for the benefits of the patients with the common diseases. And I'm going to give an example to that, but just a few general examples that you could see here. So early onset dementia, which is, thank goodness, a very rare disease, helps shed light on Alzheimer's disease, which is extremely common. Rare monogenic autistic spectrum disorder helps shed light on autism, sadly not a rare disease at all. And I want to give an example for the TEVA pipeline. So rare pain disorders can serve as a window for common pain disorders. And there are 2 of them. So one is Mendelian pain insensitivity disorder. This is an extremely rare disease in which deems the patient completely insensitive to pain. You could see here an example. There was in the New York Times about a patient. So during the interview, the patient has taken boiled egg out of boiling water with her hands. So these patients suffer various types of injuries. They could sometimes bite off their tongue while eating. They have no sensitivity to pain. They have all other sensitivities. And life expectancy is reasonably low in these patients. So it's a really devastating disease. And it's a monogenic disease. The gene behind it codes for a protein within the NEV17 ion channel, which mediates pain in the periphery. The ion channel is non-functioning, and therefore there is no pain sensitivity. The gene was identified in 2006 by a group in Cambridge working to identify very rare diseases, and in that case they identified it in a Pakistani patient living in Iran working as a street performer because he was insensitive to pain. So he would work on hot coil, he would swallow fire and knives, and so on. It also shows you, I think, something about the importance of collaboration. Somewhere in this world, we have answers for a lot of our problems. We need to be outgoing and to outreach to the right places to find them, even in unexpected places. The second disorder is a disorder leading to a disease called erythromegalia, which is the exact opposite disease. So it's a disease of extreme sensitivity to pain. There are images, how the patient describe how they feel, and they describe constant burning sensation. For example, from light touch that we would experience as touch, they already experience as pain. And what we experience as light pain, they experience as extreme pain. There is suicide rate in carriers of this mutation, which is a gain-of-function mutation in the SCN9A gene, the same gene from before. And in that case, the gain-of-function mutation leads to hyperactivation of the NEV17 ion channel, meaning that every touch that activates the ion channel is mediated much stronger, so more action potential and more channel opening. So you see really that these are two sides of the same syndrome and actually mediate through the same mechanism. This was identified also in a very rare family in 2004. So if you bring these families together, you kind of have this question, can you see a spectrum? And are we, all of the people in the room, on the spectrum? And the answer is yes. So on one side, we have the extreme pain insensitivity, and you could see here an example of another patient also working as a street performer because of his pain insensitivity. On the other side, we see erythromegalia patient with extreme sensitivity to pain. And in the middle, we have the entire population, which also has polymorphism for the SCN9A gene, making them different in their pain sensitivity. As many people in the room can attest, also people without diagnosed diseases have diverse difference in pain sensitivity. So if you put this together, what you have is actually a novel target for treating pain, because you could look at this general gene variance and ask yourself, can I treat it? And by that, create a new medication for pain, an extremely common syndrome that is of need of treatment, because current treatments are usually opiate-mediated, so very addictive. So a disease in great need of care. And the answer is yes. So TEVA has been developing a drug for pain based on this mechanism. And more than that, we could go back and look at the polymorphism of the general population and see within it how are people going to respond to the medication. So this is the first step toward personalized medicine. So all of this is an example of how rare diseases can teach us about common diseases. But this is a little easy because I stopped at the point that we identified the gene and we identify the ion channel that's affecting it. In reality, drug development is a lot more complex than that. So just kind of the end product of the complexity is that most clinical trial programs will fail. You could You can see here the breakdown across diseases, and you can see that overall only 1 out of 9 programs will actually reach the end. And these are programs that have already reached clinical trials. A lot of them will fail earlier than that. So we want to understand what can we do about this. Not only find new targets, new genes, but also find how to make something meaningful from this target that would reach all the way to the end and all the way to the patients. So what do we need in order to make it from a target to the market? So we need the target validation, then we need to identify hits for compounds that could affect this target. Then we need lead optimization, so we need to optimize these leads to make them chemically potent. Then we need preclinical development, which is nice to put it in one box, which is, of course, has many components, for example, efficacy, toxicology, metabolism, and so on. Then, assuming that all of this worked in animals, then you have to go through clinical development, phase 1, 2, 3, and so on. And in the middle of this, we have what is called the valley of death. So we see a lot of interesting things in this area. So a lot of interesting work, a lot of interesting academic work, a lot of interesting industry work. But very few things actually make it to clinical trials, and very few things make it to the end of the clinical trials. Why? One way to put it is that we have on this side academics, and on this side we have industry. And the culture is very different. So academics don't have the funds and don't have even necessarily the interest in this part of the equation. And industry has the funds and has the interest, but it cannot risk projects that would fail because of this great investment. Another way to frame the exact problem is that in between this part and this part, we have a predictability gap. So it's very hard for anyone to predict at this stage what would make it to the end. This predictability gap is because we don't have good translational models. So from what we find here molecularly and in animals to what would work in full live patients, there is a huge gap of translationability. Part of it is to understand efficacy, to understand metabolism differences between animals and humans, to understand toxicology and problems that would emerge in patients, to understand how to make a drug potent, specific, and selective, which might be very contradictory, and to understand how we could develop biomarkers to allow us to understand how the drug works as well as good as possible. And just to give an example from rare diseases, it's a sad example, it's a field that we work in, and that's the therapeutic effect of Huntington's disease mouse models. So this is an example of different potential therapies for Huntington's disease that were tested in different mouse models. And you can see here this wonderful quote. So because they are actually very good models at many levels of the analysis, it would be surprising if some compounds benefiting the mouse models did not prove to be beneficial to humans with HD. However, we stand now with no treatments for HD. And if you look at this exact list again, almost all of these compounds have failed in the clinic. So as good as these models were, they were not good enough to be translational. And this is exactly this translationability gap that I mentioned before. We need to understand how to go from preclinical to clinical effectively. Now, if you look at rare diseases, this problem becomes more severe. And the reason for that is that people are themselves very diverse. And people diagnosed with the same disease don't have the same disease, they don't have the same phenotype, they often don't have the same genotype. Most diseases are not monogenetic, and there actually are not one monolithic group, but many groups. For example, some people apparently feel that the air conditioning in this room is pleasant, where other people find it freezing. So just as a good example for the differences between people. So this difference extends to other diseases, such as pain sensitivity. And the problem is that rare diseases are as complex as common diseases. As diverse and as complex. Most rare diseases are not monogenic. Most rare diseases have a huge difference in manifestation, and we need to address this difference if we want to cure them. And in order to do this, we need to have insight into this diversity that we barely have for common diseases. So how are we going to have this insight for rare diseases? We have less patients, we have less understanding of them, and maybe we have less ability to look at phenotype, we have less ability to look at genotype, we have less population to even test a potential treatment on. So we have all the other challenges, and we have this one. So what can we do about it? What can we do to solve it? So I'm going to talk about 3 answers. The first of which is data. So one thing that has changed and is changing every day right now is the availability of data. We have the ability to collect, store, aggregate, and analyze data. All are improving any day now. And this is not only genetic data that has been mentioned a lot today, but also clinical data, and also data that is not immediately disease relevant, but is becoming very meaningful in our understanding of diseases. For example, data from various bodily sensors, from different applications, and so on. And we could use this data, the aggregation of this data, to look at better, to understand disease mechanism and identify targets, to look at how different potential treatments are affecting our patients, and to stratify the patients into groups, because we would need to do it also for rare diseases. So just as an example here from the ALS Therapy Development Institute and their Precision Medicine Initiative. So they asked patients, this is a nonprofit that works outside the clinical institute. So they asked patients directly online to register to their program, and they provide samples for analysis and to create iPSCs and for genetic testing. So genetic testing and iPSCs. They also gave them sensors to track their activity levels and to track their speech, and to also record their functional rating scale and a questionnaire. And if you have both of these functional measures and these biological measures, you can look at all of these different things. And I hope that if we're going to have this conference again next year or in 2 years or in 3 years, we're going to see speakers here not coming only from biology and chemistry and medicine, but also people coming from computer science, from engineering, from statistics, working together on this problem and how could we understand rare diseases by utilizing new sources of data. If we're going to bring these people in, we're going to have more people working on rare diseases. One other way to approach the problem of rare diseases is new models for developing drugs. And one such model is repurposing of existing drugs. So you could see here an example for how common is it to use repurposing drug, and it's becoming increasingly more common. Right now, 30% of the drugs that are approved on the market are drugs used for other indication, repurposed for new indications. And the reason for that is that the drug development is easier, if indeed it's effective. So the development time is shorter. Repurposed drugs are already known to be effective, and you already have a much better sense of their tolerability and their mechanism. You are much more able to mediate the problem of safety and to mediate the problem of not understanding the mechanism well. So you see a lot of interest in this mechanism. It's only growing. It allows us to access new drugs that we weren't able to access them and to bring them to the patient faster. One way to do it is a data-driven approach, meaning you could try to look at the different drugs that are available on the market that you might repurpose and try to look at the gene expression associated with these drugs compared to the gene expression generated by the patients that have your disease, and try to identify a match. So this is the sort of hypothesis-free approach that was discussed here before, and it allows you to screen through many diseases and many potential drugs and find matches that you might not be found based on scientific rationale alone. And TEVA has been working on this approach in collaboration with a US-based company called Immuneering. And as of June 2016, we already looked at 12 different indications and settled for 2, which we were able to identify good and interesting data, which shows you again the importance of data. And we looked at over 100 different candidates for these diseases, and we are now testing 2 drugs out of this program. The third way to approach the problem of rare diseases is the way to approach the problem of the valley of death in drug development in general. And this is for academia and industry to work better together. So we both have strengths that we want to utilize and we will have to utilize to address rare diseases better. And you could see here, and I think all of you know this, there is a lot of benefit for academic research that need to be better translated to get to patients. So we want to be able to harness the capacity of academics with intellectual novelty, new ideas, new targets, a lot of curiosity-driven research that doesn't exist as much in industry, and to understand how we could work on that together. For example, if you look at different drugs that were developed in academia, you can see that over 30 different drugs that we use today, that are on the market today, came from academia. And importantly for this conference today, a lot of them were actually developed in Israel. So if we look at different neuroscience drugs that were developed in academia, 4 out of the 10 were actually developed here in Israel. So we are really a powerhouse of new ideas for drugs, and we want to harness that. How do we do that? For that, we need to understand also the limitation of academic research. So academic research is very successful in identifying novel ideas and developing them, but only developing them until some limit. What academic institutes usually don't have, and I think we're trying to change that here, is a good understanding of the medicinal chemistry involved, identifying good leads and optimizing them, how to develop assays and assays optimization to identify screening of a lot of compounds, to understand drug metabolism and pharmacokinetics well, and this would enable us to really reach a point that we say we actually have a potential drug. So this is something where we could really contribute, where industry can contribute. We can evaluate a project feasibility from a chemical standpoint, from an industrial standpoint. We can evaluate from a chemical standpoint the property of the lead molecules, address potential toxicology problem and other developmental problem, look at potential assays to assess the drug and to allow screening of more molecules. And this is really the question of how do we work together? So how do we create a program in which academic members utilize their strengths and industry members utilize their strengths to bring together a solution that is translatable and we actually can say strongly that it's likely to reach patients and change their lives? And so how do we do it? Well, this is kind of our internal recipe. This is what we believe would be good in academic collaboration with industry. So it needs to start early. It needs to start at the point that you're looking at the target and you're trying to identify potential compounds to fit this target. Each partner needs to contribute. So it's not one partner looking at the other, but working together, with academia providing the research, the target, and the animal models, and industry providing the medicinal chemistry, assays, toxicology, pre-formulation, and metabolism, just to name a few things. And just to say a little bit, how do we see this in Israel? So we have several programs that are already focused on rare diseases. One of them that many of these people in this room know and are part of is the Neuroscience Network of Excellence, which is a program in which TEVA is committed to supporting neuroscience research in Israel. And I just want to note that in 2016, we supported 19 projects, of which each 8 were focused on rare diseases. So we're very interested in rare diseases. We also have a collaboration with the Bar Ilan Medical School specifically focused on the rarest, extremely rare diseases in the north of Israel, because we believe that this is one of these cases that we could help patients with rare diseases and also ideally learn about common diseases. Beyond these programs, we're open to new potential collaborations according to our recipe. We would like to work together from the beginning and until the end, utilizing each and every one's strength. If there are people in this room interested in this approach, we'll be happy to talk more. Thank you so much.